The overall goal of this procedure is to image the heart function of Drosophila longitudinally in vivo using an optical coherence microscopy technique.
This method can help answer key questions in the field of Drosophila cardiac structural and functional development by providing metrics such as changes in cardiac diameter, heart rate, and cardiac activity period during Drosophila metamorphosis. The main advantage of this technique is that the optical coherence microscopy is capable of imaging small animal hearts non-invasively with high spatial and temporal resolution.
This technique may help reveal the mechanisms of human cardiac disease and the relation to cardiac development due to the genetic similarities that exist between Drosophila and vertebrates. This method can also be applied to other systems, such as optogenetic pacing, for studying or treating weakened pacemaker function in animal models.
When producing the flies for this experiment, allow reproductive parental flies to mate and lay eggs in a fresh vial for 8 hours. Thus, the clutch of eggs laid will develop into flies of a similar age. To mount a larva for OCM imaging, first prepare a clean slide with a piece of double-sided tape. Push out any trapped air bubbles below the tape. Next, use a soft brush to remove a larva of the right size from the vial. Place the larva on a clean tissue, remove any food stuck to it using a moistened brush, and then let it dry. Now, under a wide field microscope, confirm the developmental stage of the larva. Then,